Have you ever wondered why you crave certain foods? Or why you react to stress in a particular way? The answers might be buried thousands of years ago in the genetic blueprint of our Stone Age predecessors. Join us as we journey back to the dawn of humanity to connect your present to their ancient past. The ancestral blueprint unveiling the out of Africa theory and its genetic evidence from modern populations. The out of Africa theory is one of the most compelling narratives in human history. It proposes that all modern humans share a common origin in Africa, from where our ancestors migrated to populate the rest of the world around 60,000 to 70,000 years ago. This theory gained strong support from genetic evidence, especially when researchers began comparing DNA from different populations. They discovered that genetic diversity is greatest among Africans, suggesting a longer period of evolution there compared to anywhere else. This makes sense. The longer a group has existed in one place, the more genetic variations it accumulates. Studies of modern populations show that all non-African genetic lineages can be traced back to a small group of migrants who left Africa, possibly following coastal routes into Asia. Fossil records, archaeological findings, and genetic markers align to paint a consistent picture of this migration. It wasn't a single journey, but a series of waves over millennia, shaped by climate changes and resource availability. Every human alive today carries this history within their cells, a living reminder that no matter where we come from, our story began in the same cradle of humanity. Exploring the critical role of mitochondrial DNA and Y chromosome tracing in mapping human migration. To trace the footsteps of our ancient ancestors, scientists often turn to two powerful genetic tools: mitochondrial DNA, mtDNA, and the Y chromosome. Mitochondrial DNA is passed down from mothers to their children, almost unchanged, while the Y chromosome is passed from fathers to sons. Because these genetic materials change very slowly over generations, they act like molecular time capsules. By analyzing specific mutations, researchers can identify haplogroups, genetic families that share a common ancestor. For example, mtDNA studies have helped locate mitochondrial EV, the most recent woman from whom all humans today are descended on their maternal line, who likely lived in Africa about 150,000 to 200,000 years ago. Similarly, Y chromosome studies reveal Y chromosomal Adam, a paternal ancestor who lived around the same time. These genetic signatures allow scientists to map how different lineages moved and branched across continents over tens of thousands of years. Through these tools, we can see how people adapted to new climates, encountered other human species, and developed diverse cultures. They provide a direct genetic link between modern humans and the courageous migrants who ventured into unknown lands. Shaping the world we know today, the latest discoveries on interbreeding with neanderthals and denisovans. How much of them is in us? For a long time, scientists believed Homo sapiens completely replaced neanderthals and other ancient humans without mixing. But genetic research has rewritten that story. When scientists sequenced the Neanderthal genome in 2010, they were shocked to find that most people outside Africa carry between 1% to 2% Neanderthal DNA. This means our ancestors interbred with Neanderthals as they migrated into Europe and Asia. But the surprises didn't stop there. In 2012, genetic studies uncovered DNA from a mysterious group called denisovans, identified from fossils found in Siberia. Denisovan DNA is present in certain populations in Oceania, Southeast Asia, and Tibet. In some cases, up to 5% to 6%. These ancient encounters left lasting impacts. Neanderthal genes influence our immune systems, skin pigmentation, and even risk factors for certain diseases. Denisovan genes contributed to traits like high-altitude adaptation in Tibetans, while the majority of our genome comes from Homo sapiens. These small percentages remind us that we are a blend, the result of ancient interactions between different human species. Far from being separate and isolated, our ancestors lived in a web of connections, sharing not just territory but also bloodlines. Echoes in our genes how ancient environmental pressures like ice ages and plagues shaped our genetic predispositions. Human evolution is deeply shaped by the environments our ancestors endured. During the ice ages, survival required genetic adaptations to extreme cold, such as changes in metabolism and fat storage. Harsh climates favored traits that allowed people to conserve heat and efficiently process high-calorie diets. Likewise, Waves of deadly diseases like the Black Death in Europe or smallpox in the Americas created powerful selection pressures. Individuals with natural resistance to these diseases were more likely to survive and pass on their genes, slowly shifting population genetics. Even today, some populations carry genetic markers tied to these ancient survival challenges. For example, certain immune system genes common in Europeans trace back to those who lived through medieval plagues. Similarly, malaria-prone regions saw higher frequencies of traits like sickle cell, which, while risky in other ways, can protect against the disease. These environmental pressures didn't just shape physical traits. They influenced human behavior, social structures, and cultural practices. Our genetic predispositions today, whether it's how we metabolize food or respond to illness, are echoes of battles fought and won by our ancestors in the face of nature's harshest tests. The surprising genetic advantages passed down from hunter-gatherer lifestyles. Still relevant today for over 95% of human history, our ancestors lived as hunter-gatherers. This way of life left genetic imprints that continue to affect us, even in our modern world. Hunter-gatherers needed endurance for long-distance travel, keen eyesight for tracking prey, and strong immune systems to survive varied environments. These traits, once essential for survival, remain embedded in our DNA. For example, our bodies are highly efficient at storing fat, a valuable trait for ancestors who faced food scarcity, but one that can now contribute to obesity in food-abundant societies. Our ability to digest a wide range of foods also comes from this era, as hunter-gatherers consumed diverse diets depending on the season and location. Some researchers believe our natural sleep patterns, social instincts, and even problem-solving abilities are tied to these ancient lifestyles. While we no longer roam savannas or dense forests for survival, the hunter-gatherer legacy still influences our health, its behavior, and even our psychological needs. Understanding these inherited advantages and their potential drawbacks helps us navigate the mismatch between our ancient biology and modern living. Examining specific genetic traits, lactose tolerance, disease resistance, and altitude adaptation, and their ancient origins. Certain genetic traits in humans tell fascinating stories about adaptation to specific challenges. Take lactose tolerance. Most mammals lose the ability to digest milk after weaning. But some human populations, particularly in Europe, parts of Africa, and the Middle East, evolved the ability to drink milk into adulthood. This adaptation likely arose in herding societies where milk was a valuable food source. Disease resistance is another key example. The sickle cell trait, while dangerous in its severe form, offers protection against malaria, a deadly disease in tropical regions. Similarly, genetic variations in the CCR5 gene provide some resistance to HIV and other viral infections, a potential legacy of ancient plagues. Altitude adaptation is seen in populations living in the Andes, Ethiopian highlands, and Tibet, where low oxygen levels challenge survival. These groups developed unique genetic changes affecting hemoglobin production and oxygen use. Such traits highlight how human genetics is shaped by specific environmental pressures and survival needs. They're living proof that our DNA is not static, but constantly adapting, carrying the fingerprints of countless generations who met the demands of their world head-on. Our prehistoric superpowers, the genetic underpinnings of human curiosity, and our drive for exploration? One of the defining traits of Homo sapiens is our relentless curiosity. This isn't just a cultural phenomenon, it has deep evolutionary roots. Dopamine is a neurotransmitter linked to motivation and reward, and higher activity in these pathways could have driven humans to take risks, explore, and innovate. This drive for exploration likely played a role in the rapid spread of humans across the globe. From crossing dangerous seas to venturing into icy tundras, curiosity pushed our species into every corner of the planet. Today, this same instinct fuels scientific discovery, space exploration, and creative innovation. What began as a survival mechanism, the need to see what's beyond the next hill, has evolved into one of our greatest strengths, enabling us to build civilizations, question the universe, and continually push the boundaries of the possible.